So let's start off defining what metabolism is. This is the steps in which a molecule is consumed and the energy from it is used. It can be used to form building blocks which can help form the basis of life, as your metabolism, as you'll probably remember from an earlier video, is a key concept of life. However, things can lay dormant when they are not producing energy, such as in endospores. They lie dormant when conditions are unfavourable, but when conditions become favourable again, they sort of germinate and then they begin metabolising again. And your metabolism is made up of two components. So you've got anabolism, which is the formation of larger products from smaller ones, and catabolism, which is the breaking down of larger molecules into smaller molecules. I always try and remember cannibal. Cannibal eats stuff. And one thing that I'll never forget from GCSE physics is the first law of thermodynamics, which is that energy cannot be created or destroyed. It can only be converted between different forms. And one example of this would be coal. So originally it is chemical energy stored within the coal, but then this can be converted into things such as electrical energy, light energy, and many other different types. And this is all made possible because there's a flow of electrons between the molecules. A redox reaction is where both oxidation and reduction takes place. Endergonic reactions are spontaneous and they always consume energy, whereas your exergonic reactions produce energy and these can be more spontaneous. And then one of the best mnemonics ever, oil rig, stands for oxidation is loss and reduction is gain, and this is of the electrons. So if something's oxidized, it loses electrons. If it's reduced, it gains electrons. A very important molecule for living organisms is ATP, which stands for adenosine triphosphate, and this acts as the energy currency of molecules. And it's generated through the ATP-ADP cycle, of which I'll show you a diagram of in a second. ADP stands for adenosine diphosphate, meaning it's got one less phosphate. So here you can see at the top, I've got an ATP molecule with an adenosine, a ribose ring, and then 3 phosphates. Coming down from there on the right hand side, you can see that a phosphate is lost, as is energy. This converts into ADP. This energy is released because one of the chemical bonds between the phosphate releases a lot of energy when it's broken, forming ATP. However, it can be easily regenerated into ATP again by adding another phosphate, but this uses energy. And another thing is that it could also further break down by losing another phosphate to become adenosine monophosphate, and that means it only has one phosphate on it. Now all this doesn't happen spontaneously, it requires enzymes, and an enzyme is a specific type of protein that is able to catalyse cellular reactions, and without these catalysts then reactions would be really, really slow. But the main goal of an enzyme is to go from a substrate to a product. This can be through the breaking of a bond between a substrate to form 2 new products, or the combination of 2 substrates by forming a new bond to form 1 product. However, because the proteins, they can be easily denatured or changed/damaged by extreme temperatures and extreme conditions. However, there are some forms of bacteria that can survive in more extreme conditions. So, for example, those that can survive up to really high temperatures, we've been able to use the enzymes in these bacteria in processes where human enzymes don't fully function. So, for example, in PCR, human enzymes are denatured when splitting the DNA, but we can use these bacteria's enzymes instead. Enzymes can also have these small molecules called coenzymes associated with them. These just aid the function, but they aren't a substrate.